Thank you very much. So Katie and I have done this for a while, and what we usually do is she goes first, so she can give the background of how we develop nucleoside-modified mRNA. And that allows me to talk about its uses, its use in vaccines, and future uses in therapeutics, gene editing, and other technologies. So I just wanted to show one slide that Katie doesn't show. And what this slide shows is that there are 17 innate immune sensors that recognize RNA. And they signal in a variety of mechanisms. They make inflammatory cytokines. They directly inhibit translation. They induce type 1 interferons. They induce apoptosis and cell death. They make animals sick. And this is the problem that Katie and I had when we first started working with RNA. When we injected the RNA, the mice got sick. So I just want to recap what Katie talked about that we discovered and developed with nucleoside-modified RNA. The first was that it doesn't activate any of those RNA innate immune sensors. So what that means is it doesn't make inflammatory cytokines. That has a couple of effects. One is that it doesn't have adjuvant activities. Vaccines require adjuvants to work well. It also allows high-level translation, and the modified RNA, when given to an animal, makes hundreds to thousands of times more protein than inflammatory RNA. So Katie and I did this work in the early 2000s, and people at the time would ask us, why are you wasting your time working on RNA? People had little interest. Biotech and pharmaceutical companies had tried siRNA, had tried mRNA, had tried other RNAs, and failed. They couldn't develop a therapeutic. So they abandoned RNA. And they would ask us, why are you guys wasting your time working on RNA? Nobody is interested in it. And what I would tell them is, protein therapeutics for pharmaceutical drugs is one of the most rapidly expanding elements. And in order to make a therapeutic protein, you start with a 50,000-liter drum full of CHO cells or other cells that make your protein of interest. You then have to figure out and develop a way to purify that protein away from all of the other cell culture and cell contaminants. So every time you want to make a new protein, you have to start over. You make a new cell line, you develop a new purification. What that means is that for monoclonal antibodies, and they're probably the most rapidly expanding element of pharmaceuticals, the cost of a monoclonal antibody is $2,000 to $30,000 per dose. That becomes very quickly unaffordable for many people in many regions of the world. The advantages of RNA are that it's made in relatively small vessels. So Moderna and Pfizer use 100-liter bioreactors in their GMP facility to make RNA. The purification is one step. It's identical no matter what the RNA makes, what it encodes. So you don't have to reinvent the procedure every time you make a new vaccine or make a new therapeutic. And it's also safer. The CHO cells in a 50,000-liter stainless steel drum sometimes don't make the protein correctly. And patients will have an immune response against it that can have bad effects. RNA uses our own cells, host cells, to make the protein. Our cells know how to fold, know how to modify, know how to make it correctly. So it's safer, it's less expensive, it's much easier to expand, and that's why we continued to study mRNA. Now the issue then became, how do you deliver mRNA? And the problem is that every fluid in our body and just about every cell in our body has an enzymes known as RNases. these rapidly degrade single-stranded RNA. So you have to avoid these enzymes. And the way we approached this was to wrap the RNA in different kinds of materials. We studied around 40 different materials and finally identified lipid nanoparticles as the best. So lipid nanoparticles are made of 4 different lipids that self-assemble into 60 to 100 nanometer particles. They bind ApoE, which targets them to the liver. They also target dendritic cells, which is what makes them very good vaccines. They're safe and they're very effective. So when we first started these studies, we wanted to know what happens when we inject RNA into an animal. And we did this by making an RNA that had a luciferase enzyme. Luciferase is the enzyme you see in fireflies that makes the fireflies produce light at night and glow. So you can inject luciferase encoding RNA into an animal and then give them the substrate for that enzyme and they'll produce light. And you can measure that light in a camera. And what we've done here is we've injected RNA-LNPs by a variety of routes. Anything with systemic distribution goes quickly to the liver and is translated for a day or two and stops. But what was interesting is when we used peripheral injections, intradermal, intramuscular, subcutaneous, we had local translation that continued for up to 2 weeks. That made us think this would be a good vaccine. So we started working on vaccine development in the early 2000s. This is just a recap of how you make an RNA vaccine. And what you start with is the sequence of the protein of interest. So for COVID-19, that's the spike protein. You optimize every element of the RNA, the coding sequence, the UTRs, the cap, the poly A tail. You in vitro transcribe it. That's a reaction where the enzyme makes copies of a DNA template to produce the RNA. You purify the RNA to remove contaminants, and then you add it to lipids that form a lipid nanoparticle. You remove the salts, and that's your vaccine. So when we first started this, we looked at influenza hemagglutinin. And this is just a recap of the studies that were ongoing when we started. And what they show is that many different vaccine approaches, live virus, intramuscular protein, and activated viruses, typically gave HAI titers, neutralizing titers, of about 1 to 100. That's not bad. 1 to 40 is protective. So in humans, 1 to 100 is considered a decent antibody response. So we made an mRNA that encoded the same hemagglutinin, and we injected that into mice once. We had titers of 1 to 2,500, 25 times greater than standard vaccines for influenza, 5 times greater than infection of a mouse with influenza virus. This caught the attention of biotech and pharmaceutical companies as really a potentially fantastic vaccine approach. But as basic scientists, we wanted to understand why were we seeing such an enormous titer? And this is just a recap of how B cells recognize antigen. So a follicular B cell binds to the virus, it gets help from a T cell, and it becomes a germinal center B cell. A germinal center B cell rapidly proliferates and affinity matures. So what that means is that it increases its binding for the antigen of interest to make better neutralizing antibodies. It then goes on to become a plasma cell or a memory B cell. We measured these populations in an immunized mouse. These are antigen-specific B cells. So these are B cells making hemagglutinin antibodies. and we've stained them for the different types. We're comparing to an inactivated influenza virus. And in all cases, for all three types of cells, there was about 50 times more antigen-specific B cells produced by mRNA compared to an activated virus. That explains the huge antibody titers that we saw. Plasma cells are important because plasma cells are what sit in our bone marrows for potentially our entire lives and produce antibodies of interest. So most of us, or the older folks in this room, probably have plasma cells in their bone marrow that make antibodies that protect against smallpox. The younger folks have plasma cells that protect against tetanus and measles, mumps, rubella, other common antigens. But I mentioned earlier on that the RNA that we produced had no adjuvant activity. Vaccines require adjuvants. So I apologize for the overly complicated slide, but what it's telling us is that there are many different types of adjuvants that bind a variety of sensors on dendritic cells and activate those cells. They lead to the production of signaling molecules that promote T cells and B cells to make a good antibody response. In the absence of an adjuvant, you get poor antibody responses. Now, the problem was that the RNA that Katie and I made had no adjuvant activity. It didn't make any inflammatory cytokines. It had no adjuvant activity. So we had to figure out what was the adjuvant in this vaccine. And we focused on a particular type of cell called a T follicular helper cell. Why that's important is the T follicular helper cell drives the germinal center where antibody responses are made. And these are very potent at stimulating high affinity, potent neutralizing antibody responses. So we moved to a macaque system where we immunize with HIV envelope immunogens. And what I want you to see here is the comparison. So these macaques received an envelope protein with a double-stranded RNA adjuvant. That's considered one of the best adjuvants for follicular helper cells. And we're comparing that to mRNA-LNPs. And as you can see in both cases, the RNA-LNPs had enormous more TFH induction than the best adjuvant around. So that told us that this vaccine was special and that it induced these helper T cells that promote antibody responses. We made another interesting observation. So this is what a hemagglutinin molecule looks like. And it has two main regions, a head. The head is what binds to our respiratory epithelial cells and allows infection in our lungs. And the stalk is what allows the virus to escape from the endosome and infect the cell. The head mutates very rapidly. That's why we have to make new influenza vaccines every year. The stalk mutates very slowly, and responses against the stalk are protective against a broad range of viruses. Responses against the head are limited usually to just that virus. One mutation is enough to escape neutralization. So we developed an assay, and in this assay, in this line, the hemagglutinin head and stalk are identical to what the animals were immunized with. And we see very potent antibody responses. As we dilute the serum, we don't lose binding. In these wells, only the stalk is shared between what we immunize the mice with. The head is completely different and doesn't bind to anything in these animals. And what we saw was, again, enormous antibody responses After a boost, there was very little diminution of binding. That's not seen with typical vaccines. So that means we had a very potent response against a conserved part of the influenza gene. What that means is that this is a distribution of group 1 and group 2 HAs. So in humans, H1 is the influenza virus that infects us every year. In the past, there have been a few other hemagglutinin subclasses that can infect humans. And in group 2, it's the H3 infects us. Rarely we'll have an H5, which is an avian influenza virus that can cause a pandemic. It's very distant. It has less than 60% homology to the H1s. I told you before, one amino acid change was enough to stop a vaccine from working. These have hundreds of amino acid changes. But we challenged animals that were immunized with H1 with an H5, and they were completely protected. And that's because they made a response against the conserved stock that protected them. So this is the beginning of a universal influenza vaccine, a vaccine that can protect against drifted viruses, last year's viruses, or brand new pandemics. So our conclusions from our influenza studies were that we developed a brand new vaccine platform that used modified RNA and lipid nanoparticles. We saw enormous antibody responses and protection across subclasses, which is not seen with any other vaccine platform. The mechanisms were the induction of a specialized helper T cell population that boosted antibody responses. And that those TFHs are responsible for the potent antibody response. So we had a vaccine that would protect against distant other subclasses and had incredibly potent responses. So I'm going to change topics, and I wanted to talk to you about the future of RNA therapeutics. And the first thing I have to do is just mention Penn, where Penn is a leader in the development of cellular therapies and gene therapies. Penn developed the first CAR T cells for clinical use. They've had multiple FDA-approved CAR T cell therapies. Penn was also the first place where a gene therapy, in this case for blindness, was developed. So Penn has very strong interest in cell and gene therapies. I mentioned that lipid nanoparticles target the liver, and they target dendritic cells. But what if you want to make a therapy that targets a different type of cell? What if you want to make a therapy that loads T cells, or that loads endothelial cells in the lung, or in the brain, or in other organs? Or if you want to treat bone marrow stem cells, or any other cell type in the body? You have to figure out how to get those lipid nanoparticles to those tissues and to those organs. So we developed a very simple procedure. And what we did is we added a targeting antibody or a targeting protein to the surface of the lipid nanoparticle. So what we now have is, instead of a standard lipid nanoparticle, it has binders on the surface that can bind specific cell types. When we made these and analyzed them, we saw that adding the antibody didn't change how they looked. It made them a little bit bigger. It didn't change any of the properties of the lipid nanoparticle. They were a little bit bigger. They had the same charge. They had the same structure, the same morphology. Most importantly, they retain function. So in A, this is simply binding to PCAM expressing cells. And we've added an anti-PCAM antibody to the lipid nanoparticles. So now they bind very effectively to cells that have PCAM on them. Here, they're delivering mRNA to those PCAM-expressing cells. So not only can they bind to the cell of interest, but the RNA is taken up and translated and protein is made. These are UVEC cells. They're a type of human cell. In the absence of PCAM targeting, you get no RNA expression. The lipid nanoparticles don't bind them, aren't taking up, and don't make RNA. When they're PCAM targeted, you now have high level binding, uptake, and expression. So here we're able to target a specific type of cell, a vascular endothelial cell, with a targeted lipid nanoparticle. Importantly, we then took these targeted LNPs and injected them into a mouse. And what we can see is that in the control, so this is no antibody or a control antibody, you mainly have liver expression. But when you PCAM target, PCAM is on endothelial cells. The highest number are in the lungs. And here you see the lungs are now lighting up. So half of the activity is going to the lungs, whereas before you had none. So we were able to effectively target lipid nanoparticles to the lungs and express whatever the RNA we wanted in them. My lab has a long interest in HIV, so we wanted to see, could we target CD4 positive T cells? And this could be part of a potential HIV cure therapy. The idea was that if you can deliver enzymes that inactivate proviral HIV, latent HIV, you might be able to cure people with HIV infection by getting rid of the reservoirs. Again, we added CD4 antibodies to the surface of the lipid nanoparticles. We saw both in the number of cells bound and the amount of binding, very high levels of expression of the lipid nanoparticles on the T cells. But most importantly, the RNA was translated when we targeted them to CD4 positive T cells. That was a bit surprising because T cells don't have the capacity to take up particles. The hypothesis was that by binding to CD4, which is endocytosed by cells after binding, that would bring the RNA into the cell and allow it to be translated. And that's what we saw. Again, we injected the CD4 targeted LNPs. With that, we saw expression in the spleen. The spleen has high numbers of CD4 positive T cells, but we did another experiment. We took those animals after we removed all of those organs and imaged them again. And what we saw were lymph nodes lighting up with luciferase activity. These are para-aortic and inguinal lymph nodes. So what that means is that our targeted lipid nanoparticles, when they're injected intravenously, escape from the circulation, go into the tissues, and then drain from the tissues into the lymphatics to lymph nodes, and still retain the ability to bind to CD4 positive T cells and have their RNA translated. That's the needed situation, because now we can target any cell type in the body. We switched to an easier system, and what this system does is it has a gene that has a stopped codon in front of the gene and requires a recombinase enzyme to remove the stopped codon. And we delivered that as an RNA. So whenever a cell takes up a lipid nanoparticle with RNA in it, it turns green, it lights up. We injected CD4 targeted LNPs into these animals. The IG controls, we saw very little green production. With CD4 targeting, we saw high levels of green in the spleen and in lymph nodes. What's also important here is this is an activation marker. So these are unactivated cells. So we're targeting unactivated cells, which are able to take up the lipid nanoparticles and make protein from the RNA. And that's critical because in HIV, the latent cells are resting, they're unactivated. And this way we can now deliver enzymes that cut out proviruses or mutate them so they can't function or turn them off. So our conclusions from our targeting were that we figured out how to safely and easily target lipid nanoparticles to many different cell types. What's important was that we can now add an RNA encoding anything we wanted. So it can be a therapeutic protein. It can be a gene editing program. It can be a protein that inserts new genes. The potential is enormous. So from this, we wanted to see, could we develop new therapeutics? And we went back to CAR T cells. So if you know CAR T cells, what that therapy involves is that you take a patient, usually with a lymphoma or leukemia that you want to treat. You put them on a machine that does what's called leukapheresis. What that does is it circulates blood for about 2 hours and removes white blood cells. So you're able to get a few billion white blood cells from the patient. You then take those cells and you put them in a cell culture facility and you activate them to expand them. After 10 days, you infect those cells with a lentivirus that delivers the CAR gene and makes CAR T cells. You then take those T cells and you give them back to the patient after giving them chemotherapy drugs to make room. That therapy, it's FDA approved. It costs about $500,000 per dose because you're growing cells in a specialized culture facility for 10 days. You're infecting them with viruses, a very complicated procedure. What a CAR T cell does and why it's so important is it's a version of a T cell receptor. But instead of binding a peptide, it can bind a specific antigen. So what they've done for the CD19 CAR Ts is the CAR binds to a B cell antigen that's expressed on leukemia cells. And what that does is it activates the T cell that has the CAR to kill that cell. So these cells then bind to whatever you want them to bind to and kill. And we're seeing cure rates over 60% with this therapy versus 5% in the past. So enormous efficacy. Our idea was, well, what if we can deliver CAR molecules in vivo? So instead of having to take T cells out and expand them, we deliver mRNA-LNPs that target T cells in vivo after an IV injection. So we developed a targeting strategy and a single injection of mRNA-LNPs targeted to CD5 on T cells gave us 80% of the T cells in the animal now made the gene, now had genetic recombination. So we went to a model of fibrosis. And what our idea was is that we would take a lipid nanoparticle that had a CAR mRNA in it, an mRNA that makes a CAR molecule. We'd target the lipid nanoparticle to T cells. They would be taken up by the T cells. The RNA would escape, be translated by the ribosomes, and put CAR molecules on the surface of the T cell in vivo after injection. The CARs recognize activated fibroblasts, kill the fibroblasts to reduce fibrosis. We did this and we were able to get 20% of the T cells in the mouse express the CAR after 48 hours, so very high efficiency. 80% express the CAR in vitro. We then needed a model system. So this is just how we measured activity, and it's called echocardiography. It's a sound device. It bounces sound waves through the body, and it gives you essentially slices where you can measure things. This is a heart. This is the left ventricle and left atrium, and you can measure the size and the thickness of the walls inside of a mouse or inside of a person. A normal mouse, this is what the heart looks like when it's beating. And what you can see is it has fairly thin walls, and the size of the ventricle, this inside cavity, gets very small, which means that the heart is pumping out most of the blood. When a heart has fibrosis, the walls get much thicker and don't move as well. And that's an impaired heart function. And that's why people with heart attacks with fibrosis have poor functioning hearts. We treated animals with our T-cell targeted LNPs that delivered a CAR that kills fibroblasts, and the heart function returned to normal. The thickness of the walls was reduced, and the amount of blood pumped out was the same as the controls. These are just static measurements. In the blue are normal animals. In the brown are fibrotic animals. All of the ventricles increase in size. The amount of blood pumped out is reduced. Red is a single treatment with FAP CAR T cells. The heart function returns to normal. We stain the hearts for fibrosis. Normal animals have no fibrosis. Hypertensive animals have a lot of fibrosis in red. A single treatment of T-cell targeted FAP CAR got rid of most of the fibrosis. So we were able to treat a critical and difficult disease that has no other treatments currently with LNPs targeted to T cells that can deliver a CAR molecule. As an immunologist, we also have interest in targeting other types of cells. In this case, we looked at bone marrow stem cells. Why they're critical is that bone marrow stem cells are responsible for producing all of the cells in our body, all of the cells in our blood, immune cells, red blood cells, neutrophils, granulocytes, just about all of our cells involved in immune function and life. The red blood cells carry oxygen to our tissues. So we wanted to see, could we target bone marrow stem cells? We chose an antigen called CD117 that's expressed by bone marrow stem cells. Here we see that with 117 targeting, we can deliver RNA to these cells. We then went back and we treated mice with the recombinase enzyme. And 1 week after treatment, 90% on average of the bone marrow stem cells and progenitors had genetic editing. 6 months later, 90+% still had genetic editing. That tells us is that we've targeted the repopulating bone marrow stem cells, which are the critical population. So we're continuing those studies. We've done what's called a secondary transplant. What that means is we took the stem cells out of those mice after 6 months and transplanted them into new mice, and all of their blood cells were green. The red cells, the white cells, the bone marrow stem cells. So we were able to transplant the repopulating stem cells. We're developing this for a number of diseases. The main one is sickle cell anemia. So the importance of sickle cell is that it's a debilitating disease. The problems are it occurs in about 200,000 people a year, mainly in Africa and in India. But cases are present around the world. There is no good treatment for sickle cell anemia. The United States has developed a gene therapy. It involves taking bone marrow out of a patient, treating it with a lentivirus to fix the broken gene, and giving it back. That's about $1 million per treatment. You can't treat 200,000 people a year in Africa and India with $1 million therapy. It would bankrupt the world. What we're developing is a single injection of RNA-LNPs that will go to the bone marrow and fix the broken gene in sickle cell anemia. We can easily go to Africa and India and the rest of the world and give people a single injection of LNPs. It's really no more than what we do with a vaccine. That can then be expanded to many other of the hundreds of genetic diseases that occur in the bone marrow, that occur in the liver, and that occur in any other cells we want to target. We can also deliver toxic genes to deplete bone marrow. So currently, when people have bone marrow transplants, they're either irradiated or given high doses of chemotherapy that kill the bone marrow stem cells. It also kills the cells in the stomach and makes people sick. It kills the hair cells and makes people bald. It has lots of bad effects. Here, we can maybe target only bone marrow stem cells and selectively kill them and transplant new bone marrow stem cells to cure whatever disease we want to cure. So the future of mRNA therapeutics is incredibly broad. Vaccines, we're developing 20 plus other types of vaccines. Vaccines for HIV, for hepatitis C, genital herpes, malaria, norovirus, C. diff, leishmania, cryptosporidiosis. The list goes on and on with all the disease that can be targeted. For new therapies, we can deliver therapeutic proteins. I'm not sure if Katie mentioned, but she's been thinking about delivering therapeutic proteins to patients with strokes to treat the strokes. You can deliver therapeutic proteins to just about any cells in the body. You can deliver gene editing technology to fix genetic defects. One of our future interests is the brain. So can we target brain to treat neurodegeneration that's seen in Alzheimer's, Parkinson's, and other degenerative diseases? These are all potentials for the future. So I need to thank the people in my lab and all of the labs that we work with who perform the studies that I just showed you. Thank you very much, and I'll take any questions.